Welcome to the AgEmerge podcast, brought to you by Ag Solutions Network. Your farming challenges are unique, so your practices should be too. We're here to share emerging ideas, build connections, and provoke conversation.
Get ready to improve your soil, your crops, your livestock, and your family's livelihood. I'm your producer, Kim Chase.
And I'm your host, Monty Bottens. Thanks for joining us.
Thanks for joining us. Today, we're excited to welcome Adrián Ferrero. He is the co-founder and CEO of Biome. With recognition from the Spanish government, Illumina Accelerator, and the MIT Technology Review, Adrián is a self-described entrepreneur on a mission to improve the world. And he's doing just that as he and his company work to have an impact on how we look at and utilize the soil. He says this work, at the end of the day, helps to improve the way we are farming, the way we preserve and restore the life of the soil, and also create awareness on the relevance of the biological pathways that are in the soil. Adrián and Monty have a great conversation, so let's jump right in.
Welcome, everyone, to this episode of the AgEmerge podcast. I'm excited to be joined by Adrián Ferrero. He is the co-founder of Biome, and a lot of you have heard of Biome. And in fact, if you were at AgEmerge, our second one that we did in the conference in Monterey, you would have seen them pitch their company at that time. And they've come a long way since 2020. Many exciting things. They did win our soil health award there of all the pitch companies that we had. And we're proud to see that they're growing and developing. But it's been a wild journey. And Adrián, I just would love if you would share some of your story and how you got started and what Biome has gotten to today.
Well, first of all, thank you very much for the invitation to join the podcast. It's my pleasure to really contribute with all the contents and the story and the knowledge sharing that I've been gathering all the time since we started. We started Biome back in 2015, so 7 years ago, almost 8 years ago. So it's a long track for a startup and a lot of learnings in the way. And huge evolution, but what is the most interesting part is that the mission remains the same. So we still want to have an impact on how we look at the soil and how we utilize the soil, no? Because we've been using soil since ever, but now we are in a momentum where we can really analyze and understand the soil from a completely different perspective, more beneficial for our economic activities, but at the same time, more beneficial for the future of our planet. So how do you want?
Yeah, how did you yourself get interested in this soil health, basically analysis or quantification? I mean, how did you find your way into this approach?
Well, I'm bachelor in economic science and also expert in innovation, funding for innovation. And also I did a master in environmental technology. So the connection with the environment, nature, it's been a while. But what is more interesting is the origin of the two founders of the company, Alberto Acedo and myself. We're originally from the northwest of Spain. It's an agricultural area. It's called the Spanish Midwest because corn, sugar beets, and wheat are the most common crops in that area. So we were fully aware of all the challenges that farmers are facing when it comes to farming itself. And from time to time, we were not able to drink the tap water because the load of chemicals in the water, in the tap water, so in the system. And it was mainly because the way farmers were fertilizing. And we realized that there was some gap to really understand and increase the efficiency of the farming operations, not to have a huge negative impact in human life, no, let's say, from that point of view. And that was kind of the origin. It has been all the time there. So the first company Alberto and me built was a genetic diagnostic center focusing hereditary diseases, benefiting from a technology called DNA sequencing. We were the first specialized DNA sequencing center in Spain. It's mainly hereditary cancer. And when this company was growing, we decided to sell the company and do something that for us was more exciting, applying the personalized medicine concept into agriculture. And the right place to start this venture was California. And we have the opportunity. We were invited by Illumina, the largest genetic company in the world, to join their corporate accelerator program. And in May 2015, Alberto and myself did the backpacks and came to San Francisco. And that's how we started. The idea at the beginning was to utilize one of the most interesting bioindicators, biomarkers in nature, which is the community of microbes populating the soil in this case, because we realized that in agriculture, we were not using many bioindicators. And we knew how to generate valuable data from this community of living beings, and then how to turn this data, these profiles of microbes, bacteria and fungal species, into something meaningful and useful for agriculture, which are functional profiles of the soil. So we started developing the technology, first the analytical part, second the computing part. We applied it specifically to one crop, which is really important for California, vineyards, especially because close to San Francisco we have Napa, Russell River, Sonoma, so really nice wine regions. And in 2018, we started the process to expand our knowledge to other crops. And that's how we launched our current solution called BeCrop, the BeCrop platform, which at the end of the day is a soil intelligence platform, because we provide insights on everything that is happening in the soil. Nice journey, right?
Oh, yeah. And I think it's interesting that you started with the DNA sequencing in humans and looked at, okay, how can this apply to other areas? And did you really know what you were getting into when you started looking at the soil? I mean, as far as the quantity and diversity of microbial community when you were first, hey, let's go do soil. How hard can it be? And then all of a sudden you open up this unbelievable, I would assume, unknown area. Correct? Did it wind up being far more difficult than what you first had imagined?
Yeah, when you listen most of the entrepreneurs, it looks like they have a clear path on what they wanted to do. But for us, was more kind of discovery process. It was so exciting to apply this technology into the unknown universe of the soil, because we're considering what are the right place to start looking at. And actually, in the wine industry, we look from the soil to the bottle. So the whole industrial process is the winemaking process. And that confirms that the soil was the right place to focus. And from there, it's been continually a discovery process. We didn't know how difficult it was going to be, but somebody has to start, because when we were doing DNA sequencing for humans, the point is that you have a reference, which is the human genome. No, what you do is you take the DNA of a patient, your user, let's say, and you compare it with the reference, and then you start identifying all the mutations, and then you classify those mutations for those that are pathogenic, according to the literature, or those that have a known effect. Sadly, there are not much in research around positive mutations in humans. So mostly we look at the bad side of the mutations.
Right.
So in the soil, what happened is that we didn't have any reference.
Correct.
So that was really like, okay, we have the opportunity to generate the largest reference and the reference for soil biology worldwide. And that was, from the entrepreneurial point of view, very exciting. And from there, it's a discovering process, an interaction with all the users, the stakeholders, all our clients, to learn how we can deliver the highest value based on the knowledge that we generate.
Yeah, and that's like you said, you start with the human genome, you know it's been mapped, and you have good, but the soil, I've heard stats anywhere from we know 1% to 3% at one time, probably about the time that you're starting. Now that has accelerated a lot because of your work and others' work in this realm, but yeah, it's really kind of a black box. So I got to back up and ask you a little bit. When you said you're working in wineries, just trying to discover the application of DNA sequencing is what I understood. You looked at the soil plant, all the way to the bottle. I'm guessing you started to focus on the soil because you realized that the variance and the unknown is in the soil. The rest is pretty straightforward. Is that a good assumption, or why'd you decide to start with the soil?
Yeah, reality is that it's not as important what are the microbes, but how the microbes impact, in this case, winemaking. And that was the starting point. Now, when we were thinking about specifically at the beginning, I'm talking about back 2015, 2016,
Right.
on when we were generating the first data, sharing with our tested users and receiving the feedback. The point was like, are the microbes important, or it's more important what they do? And then it comes to your point, the discovery, the knowledge. Yeah, time ago we just knew like less than 1% of the microbes, because traditional microbial techniques were so limited. It was culturing microbes, and it was so difficult, but very technical and developed. But now we can really look at those microbes, identify them without having anything else but their presence. So there is not like conditionants to really know or develop those profiles. The second question is, okay, some of them have a name, others don't have a name. That's right, no? Because humans, well, are naming according to the discovery curve. Just a data point that we have right now in our database, we have identified so far 14 million of different taxonomic units. So different species of microbes in the soil. And just for you to have a reference, humans have been able to name around 500,000 microbes. So 350,000 bacteria and 150,000 fungal species. So there is a big.
Yeah, you've expanded the knowledge base so far by 28 times.
That's right. So, but is that an issue? Actually, no. With the time, we'll be able to name, and we identify the same references, the same taxons in the soil many times. So the same units are there in different places, in different crops. So it's the same species. Okay. So we might not have a name, but what we can do at genetic level is to make a correlation with what is the taxonomic species or identity, right? So imagine we are not able to say this is a cat, this is a tiger, or this is a lion, but we know it's a feline, and we know what is the role in the ecosystem. So that's enough so far. With the time, this knowledge will keep improving. And now coming back to your initial question, why soil? Well, when we were looking at the winemaking process, at the soil, blah blah. So when people drink wine, we realize that there are different values or different aspects that they consider relevant. One of them is the varietal profile, so the grape variety that provides certain flavors. Then the aging, and then what is called the terroir, the origin, the appellation. So Napa wines have specific flavors independently of the Cabernet Sauvignon coming from a different place. Yes. So these three groups of flavors can be connected somehow to the effect of the different microbes. So that's how we started to realize that the food dynamics are dearly connected to the biological processes caused by the microbes. And why the soil? Because soil was completely unknown, and because technology takes a little bit of time, and we cannot provide real-time answer, let's say, that is needed in the industry at the later process point. No, when you are at the cellar and you have a problem because the fermentation is going crazy, you have little room to react. So you cannot wait days to receive results. But in the soil, in the field, you have that possibility, because you can start even preventing what is going to happen in the next season based on what you observe now. And you can even anticipate things that are going to happen right now for that specific season. So that was the right place to start. And so exciting, because the impact of soil, 95% of our food comes directly or indirectly from the soil. So everything comes from the soil. Even we will end up in the soil eventually, right? So that's how we find out how our mission or our contribution to make this world a little bit better was by looking at the soil, developing a technology that was able to really understand and decode all the mechanisms happening in the soil and connect them to the digital world. Because when you put many layers of data together, now it's possible to really develop artificial intelligence around this and start predicting what is going to happen, even guessing before you go there. So yeah, the possibilities are amazing. And when we realize about this huge potential, well, we just went straight to soil. Forget, we still have some clients because they were so nice to us at the beginning, working with the seller, but we just do soil. So we receive around 2,000 samples of soil samples in our labs every week. So you can imagine the speed of growth and the specialization that we are having at this stage.
Then when you start receiving that much soil, you got to have troubles figuring out what to do with the samples when you're all done. You probably got some amazing raised gardens out back now. But
well, actually, our lab technicians are always asking for new freezers to preserve the soil, and it's like or the DNA. We have to keep the DNA at least 3 to 5 years, and then we discard it in a secure way. Actually, the amount of soil we need is very little compared to the traditional soil analysis, which is around 1 pound. We just use a couple of ounces. So, yeah.
Wow.
The AgEmerge podcast is brought to you by Ag Solutions Network. The ASN team is hands-on, digging in and invested in regenerative agriculture. Along with the proper plant nutrition and biologicals to boost your soil microbiome, we provide the ideas and implementation guidance to support you on your soil health journey. So stop farming the same way and contact Ag Solutions Network today at asn.farm.
So was there some inspiration also when you're looking at soil to the bottle? Was there inspiration because that then required the bottle to the glass to the founders? I mean, was there a lot of wine drinking involved here in the original startup process? I think that could be a key ingredient.
Well, you have to let your imagination fly sometimes. No, actually, for us as Spaniards, wine is really a cultural thing. So it's not about drinking like the alcoholic piece. I wish wine will have less alcohol so we could drink more because it's quite an alcoholic drink. But beyond, besides that part, as a cultural piece of Spain, and I will say California, and everybody likes wine. Sure. Yeah, understanding the connection between the plant growth and the food. In Europe, wine is considered food. It's not alcoholic beverage. So for us, it's like, okay, anything we eat at the end of the day comes from the soil and then has a connection to the earth, to the life, and to the microbes, right? So that was really inspiring. So developing and being able to help to make more clean, sustainable, efficient crops, because at the end of the day, agriculture is a business. No, it has to be a business and a profitable business. And if we are not able to help farmers to take advantage of the natural soil fertility instead of damaging this natural soil fertility, then probably we are not doing the right thing.
That's a good point. That's a very, very good point. Everything we do consume is a food of some kind, and we need to make sure that fuel is as good as it can be and as clean as it can be and the best that it can be for us. So you mentioned there, prior to your work, there was roughly 500,000 organisms identified. You've expanded that to 14 million. Do you have an idea of the universe of organisms? Have you projected that out of what that could be? And how has that universe of microorganisms grown in size as you discover more? I imagine it started, you thought it was this, and it's just gotten bigger and bigger. As you map more of it, you realize there's more to map.
Yes, and crop by crop as well, because for instance, when we talk about corn or we talk about vineyards or we talk about wheat, we're starting to reach that point where the discovery curve does not grow as much. So there's some potential, but then we need to increase a larger amount of samples, so hugely the number of samples to really increase the discovery curve. At the beginning, you can imagine it was crazy. Every new sample was a almost full discovery. But right now we can say that we have really good understanding on what is the biological status of the soil. I mean, who are the players in the soil, and also at functional level, because this is very important. This is one of the characteristics from our company. It's not just about profiling microbes. I mean, there is value there, but still it's difficult to really extract the juice, the value out of these microbial profiles. And everybody's pitching like good microbes, bad microbes, but what we've realized is not about the specific players, it's about how they interact within the whole community of microbes that are there. So the ecological relationships of this community. And this is what is really important. Let me just give you an example. When you compare the Amazonic rainforest with an Australian rainforest, from the taxonomic point of view, they are completely different, right? The species that you find, plants, animals, everything is completely different. But when you look at functional level, what are the biological processes happening there? The respiration of the plants, the predation of the different animals, the ecological relationships, the functions of these ecosystems, they are exactly the same because they are replicates of ecosystems, right? So this is the approach that we are bringing to agriculture, and provides a holistic picture of what is happening in the soil and how we can really extract value, because it might happen that the way corn is cultivated in the Midwest compared to how the corn is cultivated in India might be different, but the plant, and well, now that we're in a global world, the seeds probably are the same or very similar, and the issues that they are facing as farmers are pretty much the same, and the inputs that they are adding, the different biologicals or fertilizers, are very similar. So understanding what are the differences, the singularities, the specific needs, the personalized needs of this is what we're currently working on and helping, and also from different perspectives. On one hand, we're working with agronomists, the prescribers, let me say the doctors of the agriculture, right? The agronomists, the retailers, the people who is really helping farmers to understand what they need and make an accurate diagnosis, on what is happening and what could be the most efficient solutions to apply it in a specific field. So we work with them, and we have a program called BCAs, which is similar to the CCA or PCA certifications, connected to biological certified advisors, people who really know how to help at that biological level. And that was a missing piece. And we created this program to share all the knowledge that we have been gathering so far. And we welcome everybody because it's a free course. I mean, you can just get it. There is some requirements. You have to complete all the contents and also, well, make an exam at the end of the course. But this is how we're really creating this awareness about how to manage more efficiently the soil biology. And regenerative agriculture is helping a lot, because this, I don't want to say movement, because for me it's something higher. It's a way to really understand agriculture. It's not just a group of people doing specific farming practices. It's more about how we are looking at agriculture from a professional point of view, but at the same time from I don't want to use the word like spiritual or any, but more practical, pragmatical. So what is the impact, the different impacts that the farming activity, the agriculture is having at different levels. And the role that goes beyond just providing the food, because it's also preserving the capacity to produce food in the future. This is really important. When we read this report from the FAO that we've lost 1/3 of the arable soil in the last 40 years because of the farming practices, that's so painful, because that means that, I mean, the land we have is so limited, and we don't want to destroy forests, and we don't want to take forests away just to be able to grow more food. We want to really preserve and restore the natural soil fertility that has been there to help us since many years. So this is the work we are doing with the farmers and the agronomists. And on the other hand, we are also working with the input manufacturers, the people, the companies developing solutions, new solutions for agriculture, because beyond the current solutions that we have, based some of them on chemical fertilizers and so on that we've been using for a while, the natural solutions, the natural inputs that the farmers are also using, there is a new generation of inputs. And well, you have host many people that are working in this space, developing probiotics and prebiotics for the field, which are more advanced inputs that contribute in different ways to the soil and to grow the plants. And we are working with them to really understand how this solution works, what is the impact, the effect, the real effect. So they can also build functional claims to the market, and they can really design more marketing strategies or put their solution in the places where it's going to work the best. So this work, at the end of the day, helps a lot to improve the way we are farming, the way we preserve and restore the life of the soil, and also create awareness on the relevance of these biological pathways that are in the soil, because it's the first time we are able to identify, so look at them, but also measure from a quantitative point of view. And because we've been working on this technology for many years, and we're building this reference, we know what is a regular and normal situation in a specific crop, what is exceptional for good, and what is exceptional for bad. Good or bad is a concept also that is not so accurate, no? We always say high or low, because a low situation might be very good, depending what is your target as a farmer, no? Or what is the goal of your crop, the crop you are growing. So there is not many people criticize us because we use a color palette for our reports that goes from yellow, low, to blue, high. And they say, no, we want red and green, good or bad. And it's like, no, that's not the approach. Sorry. Having a very active, very high pathway of nutrient mobilization, nitrogen mobilization, for instance, might be good or might be bad, depending what is the balance of the other nutrients. No, but at least you know that whatever you are doing in the soil, plant is getting the nitrogen very quick, because the pathway is very active. Another thing is there enough nitrogen in the soil? Well, that's a different question. It might be, but traditional tests answer this question. What is happening with the potassium, with the phosphorus, with the ferro, with the calcium, with all the carbon? No, carbon is a really trending topic right now. So all of this is like the way we are really helping to connect to the soil life. Sorry if I'm taking too long and so many contents.
No, that's great. There's lots of things to discuss there. First off, I really appreciate your holistic approach. I think that's probably an accurate word that you were looking for there earlier. You know, looking at not only the soil but the community in larger. And I think that looking at that whole microbiome community and its functionality with itself is a real key, because we get too focused on individual just in one thing at a time versus looking at functional groups, and I think that's a key point there. So when you're looking at, really, we had a couple things there. You're doing the product testing for efficacy, but also maybe for application timing, application rates, and those kind of things, just trying to figure out how biological products work, because sometimes it is very frustrating to figure out the correct, you have something, but figuring out the correct way to do it is the key. And then the second thing is the on-farm testing. So farmer wants to get a little better idea of what's going on in his soil, might have a new farm, might have a trouble spot, or just might want to benchmark what he's got going on. How does he or she go about the process of getting their soil analyzed? they pull it, and then I have one central lab, or you've licensed to other labs to be able to do things? I know Lance Gunderson Region Ag Lab is doing a lot of work with you. What is that process look like? And then when they get the information back, what kind of changes are farmers making as a result of the information? Because there's two things in traditional soil testing. Now I'm rambling on here, but first off, there's a lot of soil tests have always been used to sell something in the past, right? You don't want to take a soil test because you're going to have a bill for something, right? So, and then the other thing is people are a little leery of how accurate are they? What good does it have that level on there? And there's just kind of this disbelief of they're just trying to sell me something. So, address what does a farmer do with the information that they get back from you from, doing a sample with the BeCrop platform?
Well, 4 questions.
Yeah. I'll just keep making them up as we go here. No?
Yeah, that's perfect. So let's go with the one that I will say is the easiest. You say that the traditional soil testing usually is done to sell something to the farmer, no? It's like how much fertilizer, whatever. So that's right. It's curious to see, for instance, in Europe, now there is a new law coming, and the good thing is that here in the United States, that has been done for a while already. So the one selling the inputs cannot be the one recommending or prescribing the inputs. They have been separated. So the consultant, the agronomist, and the retailer has to be different. And this is happening, and this is going to create some troubles over there because they are used to really work together, and that's not efficient. And the reason because they are splitting this is exactly because of that. Because if I'm the doctor and the pharmacy, it doesn't work well, no? That's the first thing. So the good thing is that the world is changing, and here in the States, we already have professional agronomists advising farmers. And I was at the beginning really surprised when I was trying to incentivize people like, hey, why don't you use our tool? And they were like, we will use it, but I cannot get any benefit, any discount or anything, because I want to really show my client that I'm independent, which is great. So I was like, okay, that breaks a little bit my market conception dynamics, right? That you have to give some benefits to certain professionals in order to stimulate them. For them, it was like, no, regular price. I don't want any incentive. So my client really see me as an independent and neutral consultant. Love it. So that's happening. And in this, but one of the problems we're facing is that the knowledge they have on soil biology is very little. When you go to any agronomy school, actually there is not much but a couple of courses that, don't go very deep. So opening the knowledge that we have, this is how we're able to really, help them to upgrade their knowledge and be presented as soil professionals or at the biological level. Okay, so traditional soil testing has been, and also there were really nice Excel spreadsheets, with the data where you input certain parameters and then you get the amount, the tons of fertilizer that you need. Here, the goal is a little bit different, because sometimes the decision is not just to apply something. Sometimes it's stop applying something and changing for another kind of input or another kind of practice. That's another thing, because now we're also testing different practices to learn what is the impact. I mean, everybody have heard about cover crops, and this is not applying an input, it's putting cover crops, different seeds in the field to really, improve the balance of the soil biology in the field. So that's the first question. Then you mentioned accuracy. That has been a key question for DNA sequencing in all the ways, because in human health, there are some parameters that you have to bear in mind whenever you do a DNA sequence testing, but still there is not like a certified protocol for this. In agriculture, you can imagine it's pretty much the same. When it comes to DNA sequencing and how it's done, anybody can claim they do DNA sequencing. Wow, so open. But it's a very detailed and very, fine process that at the end of the day, the goal is to generate data. But the quality and the amount of data are the key. Not more data are better than less data, but more accurate. So finding that balance is important. We are working with international organizations. Actually, Alberto Acero, the other founder of the company, is the chair of soil biology in the International Union of Soil Societies. And we are working to really promote a standardization of the analytical process. Because something we wanted to do from the beginning was not to have this for us, and just limit the scope for biomakers. We wanted to open this, and more people doing this for us as a company, setting a standard on how to look at the soil life was good. So we started to build a network of labs a couple of years ago, and that's how Lance Gatterson, Rienac Lab, and others like Waters Labs in Georgia started to really consider the option to build a lab for DNA sequencing, a metagenomic lab for analyzing soil biology based on the BeCrop standard, or the standard that we are using at BeCrop. Let me say more accurate on how we define things. So we have shared all our know-how and the lab with them. They have built the lab and they are just processing the sample. What happened in the lab is the digitalization of the soil biology in plain words, so everybody can understand. So this is DNA sequencing, digitalization of soil biology. And then our computing solution, our computing system is connected to their system, receive the data, process the data, and deliver functional profiles for all the samples that they process. This is our business model with them, and it's working really well because currently we have labs in France, in Portugal, in Morocco, in Central America, and 2 additional labs in US beyond the lab we have in California. So total 6 labs operating the BeCrop standard workflow, which is great. And that gives also clients comfort, because what happened with traditional soil testing is that there's no way to compare results. And this is bad, because then you cannot really learn something. You just learn from yourself, so you learn very little. But when we put all the knowledge together, and then we start asking questions to this data, then we can really learn as humans. And this is how powerful the computing technologies are. And we are not vulnerating any privacy or individual data, because what you want is to really understand how we can do better when it comes to the soil and crops, right? So everybody is protected and everybody's doing better progressively because we are learning all together in the same direction. And this is something we love.
Yeah, and as that database grows, it'll just continue to add more and more value to everyone participating in that. That's the beautiful part of that shared information.
Yeah, something we are going to start doing is to share openly insights on what we have found out. Okay, so kind of aggregate data on what are the most relevant disease pressure that we find crop by crop in US, per states, certain regions, because we are now accumulating enough data, so nobody recognize their own data there, and also because we're questioning the data from an anonymized and aggregated point of view. So that's perfect, and everybody learns, and this also helps to optimize the kind of practices or inputs that people is applying. We might not need to apply fungicides all the time. If we see that for this season, the disease pressure on fungal species for the most important problems that might affect any crop and a specific crop, well, probably you can take the risk, and especially now that we see the prices of the different inputs skyrocketing very high, right?
And the beautiful part is you can start to discover certain organisms. Everything in nature is a food source.
Mm-hmm.
So you can discover certain organisms that find that pathogen as a food source, and you can realize that, there were certain practices that derived this, that supported this beneficial, to combat the pathogen, or there's certain inoculums or something that you can do to where we can try to prosper life in a balanced way instead of poke the easy button and spray a fungicide, because when you spray a fungicide, not only do you kill the bad guys, you kill a lot of the good guys, and there's more good guys in the fungal world than bad guys. So, it's now we can really get, instead of it's really, smart bombs for pathogens instead of nuclear bombs. Is what the capability is. Once you have the knowledge, then you can act accordingly.
And also keep in mind that this knowledge help the input manufacturers to develop more targeted solutions, because the main problem was that you can not develop a customized or a target solutions if you don't know what you are targeting, right? So that's really important. And also genomics, and this is the reason because there are so many biotech companies also into the input world, helps a lot to design solutions that are targeting specific species, because if you know the whole genome or a specific pathogens, no? Because at the end of the day, most of the problems come from there. You also asked me about how a farmer can benefit from this kind of technology. Well, the process is so simple, because for them it's similar to the traditional soil testing. No, you have to test the soil in a slightly different way, but pretty much the same, smaller amount. So you decide what is the management unit, so the area that you manage under the same practices with the same crop and similar weather and geographical conditions. So this could range from a couple of acres to 30 acres, depending on the crop. So if we go to the Midwest corn, probably management unit is quite large. So we're going to collect a sample by collecting different cores into the same tubes, so we get a representation of the microbial community in that soil. We don't want surface, we want a little deeper, like 2-4 inches deep soil. This is where the most interesting community of microbes live. And you just send the tube that is really small. Actually, I don't have any tube very close now. Anyway, so it's very small tube. You ship it to any of the labs operating the BeCrop Technology. I already mentioned Waters, Regional Lab, or Biomakers, any of them. And like 2 weeks later, you receive this really nice report containing all the functional pathways that we've been able to identify. What we've learned is that there are 4 dimensions that are relevant for the community. One is nutrient mobilization. So everything that is relating to the nutrients, plant nutrients, and what is the pathway to enable the transition of this nutrient towards the plant, so the plant can suck directly the nutrient from the soil. If you have one of those pathways blocked, means that the plant is not getting that pathway because different reasons, mainly competition. The second one is disease risk. So what is the risk of having certain diseases? And we look both at the pathogens and also the biocontrol species, so the police and the thieves, no, let's say. Because when you measure the risk, just the presence is not enough. The presence is a trigger, but having a high risk is not just about having the pathogen there. You might have the pathogen, but the pathogen is a really good ecological balance. Mm-hmm. So it's not able to really create a risk, but the pathogen is there. So there is a slight risk and you have to pay attention. And this is pure prevention, which is amazing. Some people also send us samples because they already have a problem. Sometimes we're able to say, well, we'll see what is the pathogen specifically creating that. But usually if you have the problem, the problem is there, you have to react. This is more for prevention. So before the problems show up, or when you start guessing that something is going on, then that's the right moment to do it. Then the third dimension is stress and hormones. So how the plant is going to be stressed, which kind of hormones, how it's going to grow, the feature of different the fruits and so on. This is a dimension that microbes have also a really important role, and we can measure it. And then the last dimension, and not least, is the overall quantification of the soil quality at biological level, how alive your soil is, no, in essence. And this is a reflection of the farming practices that you are doing. Actually, we have a specific solution called BeCrop Rate that is able to rate the intensity of the human intervention in the field. Let me say in other words, how sustainable your field is based on what you are doing there. So if you put a lot of energy, a lot of fertilizers, you do a lot of interventions in the field that are not beneficial to the biology of the soil, so the structure of the community of the microbes in the soil, then the microbes are going to reflect it. So we have make this mechanism to really measure this impact. So we see some conventional farms that are rate very high, actually, because they are really reasonable on the way they are interacting with their field. And we have also organic farms that are not doing as well as they should be doing, because, well, at the end of the day, organic is about laws, regulations and limitations. So everything that falls under the limitations is okay, and not all the practices are really extremely respectful with the soil biology. Just out of curiosity. Usually regenerative farms fall into the higher rate because they are really friendly, even if they are not purely labeled as organic, which is really curious. So this is how a farmer is really getting the results from us. We also help them to navigate the results and connect to the BCA community so they can have a qualified expert to help them to take action from these results. Okay, and so this is just for them.
I mean, that's an excellent way to know, today it's a matter of input control, right? Conventional versus organic. That's all it is. It's not a result base, but this is unlocking a portion, just like a nutrient density testing of the food, or this is unlocking a portion of what you're doing, the outcomes of what you're doing. And I think that's going to have tremendous value for farmers and the ecosystem in the future. And there's likely going to be additional revenue streams that farmers can participate, and this allows us to help quantify the good things that are being done, that might be better than it being the top percentile of good things being done. So that is really interesting. That the disease risk, don't get me wrong, the disease risk, the hormones, the nutrient mobilization, that is all wonderful predictive things, and we can definitely adjust what we're doing as a result of that. The soil quality parameter, that's the one that's got a lot of amazing possibilities and future to it. But, we certainly have tools today that you can adjust your farming practices today to be better. And that certainly has a long reach on that fourth one that you're talking about.
Yeah, and also this parameter, which is just for the unit that I mentioned before, it's included in our standard BeCrop report for any analytics. So we don't differentiate. No, you get the whole thing, because once we analyze, we see everything. Then it's important that you don't get lost on all the dimensions, because usually when you do this kind of tests, it's because you want to know something specific. So the best is to go straight to your interest. And then, out of curiosity, or if you have time, look at the rest, but not get lost, because at the end of the day, you can dive really deep on the results, because what I mentioned are 4 dimensions, but then we disaggregate those dimensions in different indicators, and all the indicators are the report that we deliver. And this is for the unit. The rate is for the whole farm. And the idea here was to deliver as much value as possible, because there is clearly an agronomic value on the results that we deliver. Full stop, right? But we wanted to help farmers that are doing the right thing, or doing better, or trying to do as better as possible, and be conscious about their farming practices and so on. We wanted to help them to position their crops in the market in a better way. It's curious that the BeCrop rate is right now working extremely well in Central America and South America with tropical crops like coffee, cacao, bananas, because they export a lot, and for them it's a way to really show that they are trying to do better in the field, and they segment their farmers based on the ratings that they get, and they also price the crop in a different way based on the rate that they get from their farmers. So usually for the BeCrop rate, the food distributor or the wholesaler is the main client. That was really surprising for us, because we started offering to farmers, but yeah, at the end of the day, farmers is like, well, unless my buyer is paying me a higher price, I'm not going to be able to do this. So we went to the buyer, and we saw that actually they are really, really excited about this kind of tools, and they're the ones taking the cost of the tool for the farmers, and then what we help on top of this rating system is to provide some agronomic value by connecting them to the BCA community. And it's working, and hopefully this is happening bigger and bigger, because we see so many like CPGs, corporates, food corporations talking about regenerative agriculture and how they want to incentivize that. But actually, there are not many mechanisms that they can utilize, or even to use, not to show that this is real and it's not just a nice PR that they are going to release.
Right, and that's the problem is quantifying the outcomes, and there's a great way that, and this is certainly a start in there. And plus, I'm sure you can look at certain organisms that cause that are involved in the making stable humus in the soil. So that'll be an important component of carbon sequestration. You can look at, various species that tend to help increase stable aggregates in the soil. So that's important for, water dynamics in the soil. So yeah, it's very, very interesting. So I think, you're definitely at a point now with a large enough data set that if farmers listening here, if you haven't tried this before, you need to, and take a look at what's going on there, especially for systems comparisons, or if you pick up some new farm ground and you want to kind of get an idea what's going in there. If you're going in blind, that's a great way. Anything on the recommendation real quick as far as time of year or moisture considerations, or crop considerations of when sampling should occur to get the best information possible, or how is that impacted? Because on traditional soil testing, that varies, fall versus spring, or in crop, and moisture levels and all that. You might speak to how that maybe is a little less of an issue with this type of testing.
Mm-hmm. Well, actually, we don't see such a high seasonality as we could see on traditional soil testing, because as we I highlighted before, traditional soil testing is about fertilization, and fertilization is when you apply the fertilization, which is right now, a couple of months ago, probably. And but with biology, it's a little bit different, depending on the crop and depending on the farmer. There are going to be different questions, and what we see is, well, farmers sampling the whole year, so different points along the year. For sure, at the beginning of the season is like interesting, because then you can see what is the status of the soil after the harvest. This is so what do you need to restore or to promote in order to be successful the next season. At the middle of the season, with the flowers, flowering, and so on, that's very interesting, because especially disease risk is when and stress and hormones are relevant. And then before the harvest, usually you want to have this soil quality indicator, so you can also position the crop in a different way. If you ask me, you can do it all across the year for sure, but be sure what you want to achieve by looking at the soil. It's just about optimizing the yield, improving the yield by optimizing the inputs. Perfect. So let's do it at the beginning, and probably at the middle end. A couple of times per year should be enough for you. If you're more worried about diseases, then probably you just need 1 point, which is when the plant start to be vulnerable to all kind of problems. So yeah, it could be done all across the year. That's the reality.
Excellent.
It's very interesting. Sorry, yes, it's very interesting whenever you do something different. So this is very common. Farmers do so many experiments in the field, and they usually use certain areas of their farms to test. No, so comparative situation, that's the best value that they are going to get. So if you see there is an area with really high yield and other with low yield, and you don't know why, so test both, because then you can leverage both situations and increase probably the other side, or at least identify the biological parameters that might be impacting the lower yield compared to the other one. These kind of situations are the ones that we find most useful so far.
That is great. And a lot of times when we do traditional soil testing, it's frustrating because you can't find any differences. So
Yes.
it's great to have a tool that you can. Anything else I should have brought up today while we had our time together here?
Well, again, thank you very much. BeCrop has been used for many years. We see that this is going to remain active for many more. So if people hasn't tried yet, I encourage them to at least start assessing and understanding what is the biological. We should not be afraid of this new dimension because while it might be looking a little bit complex. There are many people eager to help us to simplify the decisions because this new layer of data are amazing. So there is, you can acquire or ask or buy these tests online. Biomakers.com, you have the possibility. You can go to your retail, ask them, because we work with many retailers as well, as your agronomists, input manufacturers, pretty much. If somebody tries to sell you an input, a solution, magical solution, ask them, have you tested with some independent company that can really give me an overview? Because the picture with and without doesn't work anymore, right? So yeah, don't be afraid. That would be my message.
Oh, very good. I really appreciate your time. And what's most exciting for me and your company is that really, we started the AgEmerge podcast and conferences because we saw that there was a lot of ag technology emerging, in genomics and robotics, automation, information, sensors, all these things. And we saw how a lot of it was just aiming at making conventional farming a little bit better. And we really want to promote the soil health principles and regenerative farming. And this technology is right at that intersection of regenerative farming and ag tech. And I just love the potential for it to better inform our farming decisions and really validate the soil health principle approach of regenerative farming, both in a quantitative way, to where farmers can be assured that they're making the right decision and going in the right direction and can get off of as many conventional inputs and those kind of things to move to a more soil health focused approach, and have an opportunity to see how it's changing their soils over time. So thank you for your co-founder, your entire team, and all that you've done to bring this to light, to really let the truth of what's going on in the soil be seen.
Well, I appreciate also the contribution that you do by sharing these kind of technologies and experiences all across the industry.
Well, thanks for listening. One of the things we think is so important for soil health is exploring the opportunities technology brings to the stage. And companies like Biome is just one of the great examples of how that is happening. Would you like to learn more about what we're doing to help growers explore and implement soil health practices? Well, you can check out our website at asn.farm, and there you can click on links to follow us on Facebook, Twitter, Instagram, LinkedIn, and YouTube. There's a lot of great things happening and always something to learn. Thanks for listening.